Today's dairy production systems are responsible for significant greenhouse gas emissions from livestock systems, especially related to ruminant digestion and manure land applications. Scientists at the Dairy Forage Research Center are studying how tannins added to feed can mitigate greenhouse gas emissions associated with ruminant digestion and land applications of manure. Tannins are polyphenol compounds that act as binding agents to proteins, which makes them harder to break down in the cow's digestive processes and make the manure retain proteins that are not as quickly volatilized into the atmosphere as greenhouse gases. As a part of my master's degree research for soil science and agroecology, I worked jointly with the University of Wisconsin and the USDA's Dairy Forage Research Center to take manure I collected from a tannin feeding trial to a field application to study the effects of tannin-enriched manure on soil fertility and greenhouse gas emissions. On May 15, 2014, an army of researchers applied over 2 tons of manure to field sites in the Dairy Forage Research Farm in Prairie, Wisconsin. First, the manure was prepped for application, adding water to buckets of manure and stirring to homogenize the manure's urine and feces. Samples from each bucket were then taken to determine the precise nitrogen application rate of the manure that was applied to each plot. These samples were frozen and acidified in the field. The buckets of different manure treatments were then carried out to their designated plots, with 6 treatments total, and spread from flag to flag in bands. These 6 treatments and a control plot with no manure were replicated 6 times using a randomized complete block design. After the manure was spread, the flags were removed from the plot for tillage. Flags were then immediately replaced in alignment with the manure bands. Greenhouse gas emission measurement chambers were installed by hand, using a tried-and-true method of jumping up and down on a piece of plywood to pound the anchors into the ground. After corn was planted, the anchors were pushed all the way to the soil surface, with a tractor-mounted Giddings probe. Emissions from manure were measured immediately after tillage, using a portable measurement insulated chamber lid and Fourier transform infrared spectrometry, or FTIR. The FTIR is capable of measuring a suite of greenhouse gases simultaneously, including carbon dioxide, methane, ammonia, and nitrous oxide. Measurements are recorded every 20 seconds for 7 minutes using Calcimet, a gas spectrometry measurement software. Over the 7-minute interval, gas concentrations increase in the chamber, which can then be analyzed to determine the flux of gas from a given manure treatment. The inside of the chamber has 2 small vents with Teflon tubing, as well as an entry and exit port for gases to circulate into and out of the FTIR from a pump inside the machine. Soil moisture and temperature measurements are taken with each gas sampling event in order to better understand the relationship these metrics have with gas emission levels. Soil samples are also taken from each plot immediately following manure application to determine the nitrogen content as nitrate and ammonium. Through the growing season, we continue to measure greenhouse gas emissions on a weekly basis, and soil sampled every 2 weeks to follow soil and gas emission trends over time. In 2015, residual effects of these manure treatments will be studied, looking at greenhouse gas emissions and impacts on soil fertility as a part of the long-term study on the impact of this tannin dietary feeding trial, as well as manure land application.